everybody. This is your extended family, citizen-driven research. And I just want to say tonight's program is super important. And for any of you or your friends who aren't able to make it this evening, always remember that we have a library. And this show will be recorded and be there as history will say, we challenged ALS and we beat it. It's coming, believe it. And with that, I'll turn it over to you, Indu.
Thank you, McFinn. And it's our great pleasure to invite both Merit Cudkowicz and Sabrina Paganoni to talk to us today. We are really honored to have both of them join us. And a little bit about Merit. I know both of them don't require any introduction, but just as a brief introduction, Merit Cudkowicz is a chief of neurology department at MGH and also director of Healey AMG Center for ALS. Merit is also director and Julieanne Dorn, professor of neurology at Harvard Medical School. And Dr. Cudkowicz research and clinical activities are dedicated to the study of treatment of people with ALS. Dr. Cudkowicz is one of the founders and also co-chair of NEALS. And also Dr. Merit Cudkowicz has launched the first platform trial initiative in ALS, the HEALEY ALS Platform Trial program. And we are so eager to learn more about what's happening as an update. And joining us also is Dr. Sabrina Paganoni, who's a co-director of MGH NCRI, Neurological Clinical Research Institute. And also Dr. Paganoni is associate professor at Harvard Medical School and works as a physician scientist at Healey Center for ALS at MGH. Her research focuses on clinical trials and therapy development for ALS, and she is a co-PI of the HEALEY ALS Platform Trial. With no further ado, I invite both of them to actually share what's happening in the platform trial and give us an update. So thank you. Merit and Sabrina, please take it away.
First of all, thank you for having us here again. This is my favorite meeting. I was just actually telling a few people today about this great group and to join. It's really one of the best community webinar groups that I've ever seen in my career here. So thank you for letting us be here. Sabrina's gonna start, but we really wanted a chance to update you on the platform trial, answer any questions, and I personally really wanted to address some of the challenges we had with Regimen E, just and answer any questions about that. But mostly we want to talk about the new ones that are enrolling and our excitement about those drugs and what's coming.
Great, thank you. Thank you, and thank you so much for the opportunity to present some updates. And today we're going to be sharing a few slides. It's relatively short, so we really want to use most of the time to answer your questions and really have a dialogue. If you could pull up the slides, that would be great. And really what we want to do is to basically summarize what we've done so far and where we are heading next. Sometimes, we give updates on our weekly webinars, but, it's good to also reflect and kind of share a summary of where we are and where we've been so far. So that's perfect. Thank you so much. And you can go to the next one. So again, we'll share a little bit quickly about the design and launch. I think we always have new people join this amazing community and the ALS family. And so I think it's good to reflect on why we're doing this study, what we've learned so far, and most importantly tonight, really want to focus on what's next, especially for those of you who may be interested in participating or referring someone for participation. If you can go next. So really the idea for a platform trial was really Dr. Cudkowicz's idea, and she and a patient, Mr. Healey, were thinking about ways to redesign the way we do trials to be able to do trials faster. And so Dr. Cudkowicz had this idea of looking at cancer. You may know that many more new cancer drugs have been developed quickly over the last few years. And so the idea was, how can we learn from them? What type of trial designs and innovation our colleagues are doing in cancer that we can learn from. And so we learned about this particular approach called the platform trial, which is really why now we have a platform trial for ALS. And what we learned from our colleagues in cancer is that simply by changing the trial design, we can actually make significant gains, both in terms of time, cost, and resources. And most importantly, especially for our community of people living with ALS, we know that placebo really places a burden on the participants. And so we wanted to reduce and minimize the role of placebos, and we realized that we could do that by working collaboratively. And so if you can click one more time, basically, we realized based on what we learned from others and our own calculations and simulation statistical work, that platform trials can have several advantages. So now I want to show you a 30-second video that explains what's different about this concept.
In the fight against ALS, the first ever ALS platform trial.
I'm just watching this
designed
update.
to accelerate the development of effective and breakthrough treatments for people with ALS. So what's different about the Healey ALS platform trial? Instead of testing just one drug, the Healey ALS platform trial evaluates multiple treatments at the same time. This means more opportunities to find groundbreaking therapies in a faster time frame. Imagine having to build a new dock every time a different ship came to port with new treatment cargo. This is how traditional trials work. In platform trials, there is one dock to receive all the cargo and connect the treatments to their final destinations.
And so that's how we started. In 2020, we built this dock. Or if you can go to the next slide, if you will, we can conceptualize that as a common house, a common protocol shared infrastructure. Essentially, we wanted to reboot and launch multiple drugs. And we call those regimens. So in 2020, we launched with 3 regimens. And then if you can keep clicking, you can see that despite the pandemic and other challenges, we've been able to continue to grow. And so from 2020, now going to 2023, we've been able to add a number of regimens. And again, each regimen is a drug because we have this common dock or the platform trial that we can use to test multiple drugs. So if you can click one more time. As a summary, we have one protocol, one infrastructure, one central governance. We optimized all of that to test multiple drugs. And so far, we have consented over 1,000 participants. Next slide. So what are the drugs that we put into this trial? Well, you know that there are many different causes of ALS and many different mechanisms that cause ALS. And so we, to begin, for our first group of drugs, really wanted to cast a wide net. We wanted to include drugs that target different mechanisms and then see which ones could be most promising. And the idea is to screen them rapidly so that then if we need a second trial, then essentially we could at least make the initial process of screening the drugs much faster so that ultimately we can bring more drugs to patients. So if you can keep clicking a little bit, like you see, click one more time, you see the first drug was targeting neuroinflammation, same as the second one, just a different component. The third drug, regimen C and D, were also testing different components or different mechanisms in the cell. And the one that we are currently still studying, Trialos, is working on autophagy, and we don't have the results of that drug. But what's interesting is now that for the regimens that we are enrolling right now, we're actually targeting a new mechanism. Both of them actually target the same mechanism, and this is because that's a very exciting mechanism that's really come to the forefront recently. And so, by already having the infrastructure that we launched in 2020, now we're able to rapidly add drugs and really include the most recent and most innovative mechanisms that we want to target. So we can go to the next slide. I also want to point out, and I see that in the chat, there are really great questions, and I'm sure we can take all of them today. We really wanted to bring research and trial access to to as many people as possible. And one way of doing that is to provide the same research opportunities across a large and diverse network. So right now we have a network of over 70 sites that are listed on the slide. And they're all part of the network. Now, in order to include the drug, though, they need to be activated and be trained so that they can perform all the procedures. And so not all of them may come online at the same time for each regimen, but then we'll talk about, how we can we're trying to streamline that and what other options we have for those of you who may be near a site that's not active yet or not activated for some of the current regimens. And we continue to actually receive requests from sites to enter the platform trial. As a brief reminder, in 2020, we included about 50 sites, then we added about 20 more. We're continuing to add because the idea is to increase capacity in big cities, but also penetrate new geographies. And the situation now is very different from the way trial recruitment or research access was just before the pandemic, before we launched this initiative, because before every trial was only available in a select number of cities or ALS clinics. And now with the platform trial, every site that participates in this initiative is giving access to their patients to all of the drugs that are part of the platform trial. In other words, the same regimens are available at every site, and it doesn't matter where you are in a big city or in a more rural area. All sites, do the trial exactly the same and have the same drugs. Next slide. I want to briefly explain how this works. So we have essentially a number of regimens or a number of drugs. Each participant only receives one drug. Which drug is that? Well, it depends on which drugs we're testing at the time that somebody enrolls or enters the trial. So for example, if somebody were to enter the trial in 2021, when we had 4 drugs, A, B, C, and D, they could access one of them. Right now, we have F and G, drugs number 6 and 7. So we have 2 drugs. And so those are the 2 options for people who are entering the trial right now. And the idea is that we're cycling through them rapidly. We're screening them in or out so that we can have the research progress faster. One important feature that I want to highlight is the so-called shared placebo. So shared placebo means that we compare each active drug compared to a common placebo group. And that's important for a number of reasons. Number one, that means that's by having a shared control group, we can reduce the number of people who get allocation to placebo. In other words, for every 4 people who enter the trial, 3 out of 4 will receive active drug, one of the drugs that are in the platform, and only 1 out of 4 will receive placebo because this shared or common placebo serves for all the drugs. And that's how we're minimizing placebo. If you can click one more time. I do want to mention that we're doing that because we are able to share placebo because in the platform approach, we are targeting the same population and using the same protocol and the same infrastructure for everyone who enters the trial, regardless of the drug they receive. And which is why, you can only do something like this in a platform approach and not if we work in a silo. Obviously, if you work as a standalone trial, then you cannot leverage this collaboration from different drug companies and investigators. Next slide. And so from the patient perspective, from the participant perspective, what's the experience? Well, people who participate will receive one drug. They will be assigned to active or placebo, but again, many more people go on active compared to placebo. The placebo-controlled portion of the trial, which is so important for us to tell if the drug is effective or not, lasts 24 weeks, which is a little less than 6 months. And that's kind of the shortest that we have found so far that, could give us sort of answers, because the goal of each regimen is really to provide a go, no-go decision for that drug. In other words, we want to rapidly screen in a phase 2 environment, that's the phase of this trial, all the drugs that come in, so that we can decide which ones to move forward. Now, from a participant perspective, we also have a second period in the study that's called active treatment extension. In the past, it was called open label extension, but the concept is the same. That means that after participating in the placebo-controlled trial, participants can get access to the active drug. And if you can click one more time, I just want to mention that really the primary goal of each regimen is to tell if the drug is effective or not. But in addition to the primary analysis on whether the drug has an effect on overall disease progression, we're also collecting a number of biomarkers and additional endpoints. And so the next slide sort of tells us a little bit more about this, because by doing all of these in collaboration with many scientists, many researchers who are helping us with this project, we can, and by having this shared placebo that continues to grow as a common resource that we compare drugs to, we can also do something that otherwise would not be possible in a standalone trial. We can learn more about the disease. We can develop new biomarkers. We're looking at genetics. We're looking at neurofilaments. And we're looking also at a number of digital tools, so smartphone applications that could be used to monitor participants in the home environment. And I want to mention that we're continuing to think about which new tools, whether it's a digital app or other tools, we can include in the trial. As the trial continues to grow and include more drugs, we want to include new innovative methods of learning about ALS, so that the trial can also, in return, give, provide the scientific community and the patient community with a lot of new learnings. So what are our results so far? So I told you we have included 7 drugs. So since we launched, we already had the results for the first 4 drugs. Now, the first 2 drugs, unfortunately, had a no-go decision, which means that we know now that these drugs are not a good fit for ALS. Those were regimen A and B. However, while that was disappointing, the silver lining here is that we were able to screen them out faster using fewer participants than would have otherwise happened in standalone trials. For regimens C and D, number 3 and number 4, those received some positive results, although not a complete super positive trial, but there were some signals of efficacy in this phase 2 trial, so much so that it's very reasonable. In fact, I really hope that the phase 3 trial will start soon, because we do need to get more data from a larger population and perhaps longer studies. Again, the goal of these initial regimens was to screen them rapidly, and now the companies will take them forward to phase 3. So more to come on regimens C and D, because we need more data from a phase 3 trial. Regimen E is ongoing. We will have the results by the end of the year. And then regimens F and G, and I really want to just talk for just 5 minutes, and then I promise I'll stop and we'll take your questions. But really, those are the ones that people can participate in. So just wanted to tell you a little bit more about those. The concept is very similar to what I just showed, because they're part of the platform trial, but there's a few things that I want to highlight. Next slide. So for both F and G, we have sort of the same structure of a 6-month study that's placebo-controlled with many more people on active compared to placebo, followed by an active treatment extension, meaning that everyone who receives who participates in the first period that can continue then receiving active drug regardless of what they received before. Now, F and G, they're different drugs. They're made by different companies. They share a similar mechanism. So we have dedicated webinars where the scientists that developed these drugs came and really talked to us about the technical aspects. I'm going to summarize it here. I know I'm not going to do justice to that, but so far we have had kind of a scientific webinar about regimen F, and we're scheduling one for regimen G in July. So for now, I'm just going to summarize F, and so that we're going to have a dedicated webinar in July for G. So if we can go to the next slide. So regimen F is enrolling now. It's testing a drug made by Calico in collaboration with AbbVie. And so essentially this drug targets something called the integrated stress response. So this is a key player that we think is part of the mechanisms that cause ALS. And this has really emerged more recently. That's why we have many more drugs now being developed targeting that particular response or integrated stress response. And regimen G actually targets the same mechanism, although with a different drug. The basic principle here is that when you have an overactive integrated stress response system due to chronic stress, there is persistent activation. And what that that results in is the accumulation of toxic proteins. Many of you may have heard about TDP-43 and stress granules. Those are sort of accumulation of proteins that shouldn't be there and that damage the neurons or the nerve cells. And so when you have this chronic stress due to different factors, ultimately this ISR or integrated stress response gets activated. And then there is this accumulation of these proteins, including TDP-43 aggregates or stress granules that ultimately lead to motor neuron disease. And so the goal of the regimen F drug is to undo this process and really restore the normal balance and really remove these toxic aggregates. Next slide. So again, if you want to hear more and you hear from the actual scientists who developed this drug, we have a dedicated webinar and you can use the link or the QR code to listen to that. Now, we're also excited to have started Regimen G. The enrollment just started a few days ago. And so we just published the press release announcing the first enrollment. The target is actually very similar, again, but it's a different drug. And it's important that we continue to study drugs that target important mechanisms, such as the integrated stress response. Next slide. And I want to mention that we have really an abundance of ways to communicate with the community if there's interest in learning more about the drugs, or if you want to hear more about sites. We have a dedicated patient navigator team who is a central resource for information. You can contact them at the phone number or email on the slide. And the QR codes will also bring you to some of these webinars that I mentioned, as well as to our newsletter. Next slide. I want to mention that we are really committed to sharing our experience with obviously the entire ALS community, but also with people who are working on other important diseases as well. And to this end, we've had a number of meetings with others. Really, the idea of having platform trials to make drug development better is really spreading across the neurosciences and beyond. And so many people now are consulting us in terms of how to set this up for their own disease. Next slide, and I think that's almost there. I just wanted to thank all our partners. Really, we are grounded in robust partnerships. The most important of them, obviously, is with the patient community, and really every trial participant or everyone who is supporting research in many different ways contributes to this progress. That nothing we do would be possible without the participation of the ALS community. So thank you for that. And I know I kept talking, but really I know in the meantime there were great questions, so I'll let the moderators take them. I don't know, Merit, if you want to say anything before we take questions.
No, just thank you, Sabrina, for giving that update. And I know some of you on the call are part of the platform trial, so also thank you for being part of this. I did want to address the kind of the Regimen E thing, so maybe I'll do it now, because I think those of you who know me, I've been, like you, advocating, for a number of years that every trial has an open label extension for people that were in the trial, because we'll get to why you need placebo, but still to prove efficacy, we need a placebo. And for the people in that trial, we want to give a chance for it. And so I've been a strong advocate for that. And then a strong advocate for companion, expanded access protocols for people not be eligible. So, when Sean and I were talking about doing this platform trial, and really his gift to MGH covered half the cost for the first 4 regimens, he said, you got to check out these companies, because if any of them goes belly up or pulls out, it's going to be bad reputation. I remember him saying that to me, and it's like, okay. So we set up a process where actually his company and Mass General vetted all the companies to make sure that they were financially sound, because I certainly don't know how to do that. And so we do that. But nevertheless, we did get a really unfortunate situation where Regimen E, which we're finishing now, really did run out of money for the open label extension. And so I just want to again apologize for that. We did vet them, and it was, I think, a combination of a small company in an awful market period, but nevertheless, it's not something that we would ever want to do. So we are trying to do our best to, have the people in that study go into an expanded access if we can. But I do want to just reassure that F and G are, first of all, they're committed to the entire cost of the double blind and the extension. F is owned by Google, so if Google goes out of business, then we're all in trouble. So I'm pretty confident on that one that they won't run out of money. They're going to come to July on a webinar on our Thursday to kind of reaffirm that commitment to everybody. And same for Denali, they're a very big company and they're committed. So in my career in ALS is the first time I've ever seen this happen, and I am sorry about that. And I just wanted to have an opportunity to tell that to the community and reassure that I don't think it'll ever happen again. So.
Thank you, Merritt. And I think all we can do is learn from our experiences. And, you know, you're a big advocate for us, actually. And, you know, we are in existence because of you. As a lot of them know, Merritt was also my husband's physician. And that's why she inspired us to start Everything ALS. And that's why I'm here. So thank you for everything you do, Merritt and Sabrina. So we'll just kick off our Q&A session. Priyanka and Brian will be our moderators today.
Yes. So our first question here is, will participants be able to access drugs from regimen A through D in the future?
Yeah, that's a really good question. So for C, and I'll answer, maybe we'll alternate. C, so for clean, they are offering an expanded access program right now at, I think about 20 or so sites. And also they're looking to get some additional funding to do even more as they plan out the phase 3. So that one is as good. With Perlenia, we're also, right now they're continuing the open label extension for the participants, and they are working with us and the Act for ALS to see if there might be a possibility to get funding for expanded access while they, again, work for on their phase 3 plan.
Perfect. Next question is, could you re-explain the different mechanisms of regimens F and G?
Explain the mechanism?
Correct, of F and G.
Yeah, so for F and G, they're targeting something called the integrated stress response. People think it's a central mechanism to the health of nerve cells or the motor neurons that underlie ALS. So essentially, when the cells don't have stress, that are not under stressful conditions, proteins do not accumulate. And this integrated stress response under normal stress, transient stress, can actually be a defense mechanism. But when there is persistent stress, for example, there are reasons that trigger ALS in different people. There may be many reasons. The ultimate effect is that this integrated stress response becomes overly reactive or persistently activated. And when that happens, essentially, it leads to accumulation of toxic proteins. One of them, the most prominent of them, being TDP-43, that many of you may have heard being linked to ALS. And it can accumulate in different forms, such as granules or different structures that can accumulate in the cell. And when TDP-33 accumulates as such, it will lead to neuronal damage. And so it's a central mechanism that we think underlies the majority of cases with ALS, because the majority of people with ALS do have these accumulations based on different studies. And so that's why trying to clear those accumulations is thought to be therapeutic. There's evidence for that, at least in the lab.
Thank you. Our next question is the shared placebo viable for regimens G and F?
Yes, that's a great question. Yes, so that's the benefit of the platform trials, that we can continue to use the placebos from past regimens for future ones, and we're also going to be getting data from new participants who are on placebo, which I think is important because standard of care is changing, which is a good news. I mean, with this year, with 3 new drugs approved, the oral Radicava, the Relyvrio, and then obviously the gene therapy tofersen, that can impact should impact the rate of progression. It should slow it down, but we can still use the past ones and the new ones. The statisticians can figure out how to do that balance so that we can have a lot of power to tell if a new drug works or not.
My perfect. Next question is, could you re-explain active versus shared placebo and its relation with the regimens?
Yeah, so basically the placebo period lasts a little less than 6 months. And for every 4 people that enter the trial, 3 will receive active drug, one of the drugs tested, for example, F4G, and only 1 will receive placebo. The reason we can have more people on active compared to placebo is because the same placebo is essentially used to compare to many drugs. And so all the drugs basically contribute a little bit of placebo, and together all the placebo data from different drugs, basically different regimens, get shared so that they become large enough to make a meaningful comparison. I don't know if that was the question. I hope I addressed that.
Thank you. Our next question is, what are the main differences between the drugs, and what strategies would you recommend for selecting a particular type of drug?
So in the platform trial, the people that come into the platform trial, let's say a site has 2 drugs or 3 drugs open, you don't get to choose actually which one that you're in. That is part of the platform design, that we think they're all, equivalently have the same chance of being successful. That's different from a traditional trial where, if a site has, let's say, 5 studies that are all independent, you could probably pick which one you might, with input from your neurologist, which one might be the best one for you. So here, because F and G have a similar mechanism of action, I think, for people coming in, they're both equally exciting. I mean, they work slightly differently. They have different pharmacokinetics, different, brain penetration, but they are both targeting the same biology, the integrated stress response.
Perfect. Thank you very much. So next question is, what is the process for dose selection?
So the dose selection is an interesting question. It's actually one of the most important questions to clarify as drugs proceed through clinical development. In other words, in the phase 1 or phase 2 space, one wants to really make sure that we know the right dose, and both for target engagement or knowing that we're affecting the biology, but also for efficacy, because we want to maximize and find the best dose. Now, sometimes this information is already available from previous studies, like in phase 1 studies or early phase 2 studies, that's already known. And if that's the case, then in the trial, we can just have a regimen with 1 dose only. And we've done that. In other cases, perhaps there may still be questions in terms of what's the best dose. Maybe there could be more than 1 that's a good candidate. And so we can also use the trial to test 2 dosages. And so by doing that, we can also learn more in these kind of intermediate, sort of the phase 2 study that we're running, which is really in between kind of an early phase and a late stage phase. We can learn about the dosage if there are still questions about it. So it depends on the regimen. That's why some regimens have 2 dosages and some don't, depending on the questions that we want to answer. In general, it's good to have something called dose response, because sometimes, it's good to see how the drug can affect biomarkers or our clinical outcomes at different dosages. If you see a relationship, then the data makes more sense. They become more consistent, and that's part of the kind of the learning experience of doing trials.
Thank you. Next question is, how confident are you that the FDA would grant accelerated approval if a treatment is found to be effective on the primary endpoint directly from the platform trial?
Yeah, that's an excellent question, and it's hard to be confident at the moment about FDA decisions because they've had a lot of turnover. We knew Billy Dunn very well, and he was there from day 1 of the design of the platform trial, as well as the statistician there. They've both left, so we are actually kind of rebuilding or building new relationships with the current people. Now, having said that, I think given the accelerated approval for tofersen on neurofilament that just happened, I think if any of our the regimens had a dramatic effect on that type of biomarker, as well as a clinical effect, we'd have a very strong case for accelerated approval. I think otherwise, the message we have gotten from the FDA is they still want companies and investigators like us coming in thinking about 2 studies. And in a phase 2 like this one, where it's the first study looking at efficacy, and if it's positive, to do a plan for a phase 3. However, because ALS is serious, if you have a drug that's very effective, they will consider a single study approval. but they won't commit to that upfront. But again, I think it's a game changer with the neurofilament recent approval, and we have not talked to the FDA since that approval. But we are planning to ask for a meeting with them, hopefully this fall, to get to know the new people and to ask those type of questions.
All right, perfect. Thank you very much. So next question is, have you considered microglia as a target?
I'm sorry, I'm not sure I understood the.
Say the word again. Sorry.
Microglia?
Oh, microglia.
Sorry about that.
Yeah, no, that's good. That's a great question, because there has been a lot of interest in microglia and neuroinflammation in general in the field of ALS. And I'm sorry I went quickly over the slides, but actually one of the drugs was supposed, regimen B, verdiperstat, that actually was targeting one of the aspects of microglial activation. Specifically, it was an inhibitor, or kind of would block an enzyme called myeloperoxidase, which is known to be highly expressed by microglia when there is a neuroinflammatory stage. Now, that doesn't mean that microglia are no longer a target. I mean, definitely that, that there's more research and there are more parts or components of microglia that could be druggable. It was that specific drug that was targeting one specific enzyme in microglia that didn't work. So certainly we're looking at all sorts of mechanisms, including neuroinflammation.
Thank you. Our next question is, why are platform trials not as common in the US?
I think the platform trials are new to neurology, but they're not new to oncology in the U.S. So I do think we're going to see many more of these in neurology. I know, for example, Michael J. Fox is funding a platform trial, a prevention platform trial in Parkinson's disease, and there's a stroke platform trial, a traumatic brain injury one. I also think in ALS there might be a value of different types of platform trials. Ours is one kind of a phase 2-3. But you could see a value of having a platform trial, for example, for C9 drugs, for in a particular population, or in the prevention space as well.
All right, perfect. Next question is, would the Healey platform trial consider TREGs or stem cells?
Yeah, I mean, I think those are definitely possibilities. So there are a few companies that are trying to work on Tregs in different ways, whether it's the cell-based intervention, or trying to manipulate some aspects of the normal function of Tregs. And also stem cells in general. There's different types of stem cells, as people know, different sources, different routes of administration, etc. So I think there's still a lot to learn about both cell-based treatments as well as treatments that target specific immune cells like Tregs. And I think that could be, that could definitely be a viable option if companies apply and there is a strong scientific rationale. So I think, there's a, obviously, in order for a drug to be part of the platform trial, the company needs to apply, and there needs to be sufficient data, scientific data, that warrants inclusion. There is a scientific committee that reviews all the data and makes the selection among the companies that apply.
Thank you. Our next question is, are there structural obstacles to extending the Healey platform trials to Sunnybrook, Toronto, or other Canadian ALS clinics who partner in many conventional clinical trials with MGH and others?
There are some, but nothing that it can't be overcome. So when we started the Healey ALS platform trial, we actually approached the Canadian trial network, and they initially said no to participating with us, with the exception of Dr. Genge and McGill. And I think the reason back then was that they thought it was too complex for the way their trial network is set up, and they preferred kind of single studies from industry. And this is not a secret. But they have come back to us now that they've seen that it's kind of happened, and they've heard from their colleagues in the U.S. that it's very feasible, and they've heard from people they care for that they like the idea. So we are starting to talk to them about that again. The only complexity is really that, obviously we need to translate things into French and have Health Canada approval, but it's very feasible. We'd be excited to do that.
Perfect. Thank you. Next question is, can you elaborate more on the stress response and how is this measured?
Yeah, well, basically, you know, what you're asking a great question about how it's measured, because we're learning about how to do that. And in fact, the trial will provide very important data in terms of how to measure the stress response. So there's a lot of work that's going on right now on biomarkers. I'm sure that those of you who follow this webinar series with Everything ALS have had different talks about biomarkers. And in particular, recently, when the integrated stress response became more prominent, or it was more kind of at the center of the attention in among ALS scientists, it became clear that there are some downstream targets or some consequences of the process that I described earlier in terms of abnormal integrated stress response and accumulation of toxic proteins that could actually be measured. And right now, there's a couple of papers that have just been published by collaborators at different institutions, and those biomarkers have recently been developed. And at this stage, they are available to be measured in the spinal fluid, or CSF, which is basically the fluid that surrounds the brain. And that's available for us to analyze by doing a spinal tap. So that's why actually we are really emphasizing the importance of collection of spinal fluid as part of some of these regimens, so that we can actually use some of the really breakthrough, most recent biomarkers that were developed, and they can be measured again right now in the spinal fluid, hopefully very soon in blood as well. But the trial will also serve as a way to further develop and validate these biomarkers, and hopefully we'll be in a situation where, like neurofilament, that can, you know, as you know, with the first one, it was instrumental for approval, and hopefully if we get data quickly and, you know, kind of, you know, on this large cohort, we'll be able to hopefully be in the same situation.
I was just realizing that every other question is on the science, so it's all on Sabrina, but she's doing great. She's spot on, and I agree completely. But I wanted to reiterate around the spinal tap that we, you know, we're asking people to donate the spinal fluid for a couple of reasons. One is to see if people who have more of these markers of the integrated stress response at baseline, do they respond differently? And also to see if we can measure then the change in that response, and does that also predict clinical response, a lot like the neurofilament, like Sabrina said, because we see every field that develops these shorter term biofluid or digital biomarkers sees drug development just go off the roof, because it means that, you know, in a shorter time you can predict whether your drug is going to have a clinical effect. So now we have neurofilament, but we clearly need more, and we know it's a big ask for people to have 2 spinal taps, but it's really critical for these particular drugs.
Thank you. Our next question is, for those who don't qualify for any trials, how do they find out if they could qualify for any expanded access drug?
That's a great question. I might have Sabrina answer, but she leads all the expanded access programs. Yeah.
Well, actually, I learned from you and from our patient community. You know, we started this experience with expanded access thanks to Merit at MGH as a single center. And then because of real interest from the patient community, for the reason that you're asking, you know, not everyone who would like to participate in research can do so as part of trials because they may not qualify. We decided to export this model that we had at MGH and really start to work with other sites. And so in 2021, we started a series of expanded access small programs, really funded by philanthropy, by people that really wanted to make this more available. And we have included, as part of that program, 9 sites, 9 different clinics. And now more recently, thanks to Act for ALS, which I'm sure many of you supported and helped with, and now that we have the possibility of applying for funding to the federal government, we obtained actually the first grant to have an expanded access that's funded by the government, by the NIH, thanks to the Act for ALS legislation. And so now we are launching this expanded access 25 different sites. So in order to know if you can participate, first of all, the eligibility criteria are very broad. So most people can participate. There are still some criteria related to safety. So for example, if people, some drugs react or interact with other medications. And so if the individual must be on some medications that can interact. Potentially they may not participate, but these are very small cases. You know, most people should be eligible for expanded access. And I think you can refer to the same patient navigation team that I showed earlier. They also serve as a resource for our multi-center expanded access protocols. And again, right now there is a network of 20 plus sites across the US, and we applied for more funding, and hopefully we'll be able to even increase the number of sites. And ideally, soon we hope to match it to all the platform trial sites, so that, you know, sites will be able to offer research to everyone, whether it's by way of a trial if they qualify, or expanded access if they don't qualify. So we're working hard to get to that level.
All right, perfect. Thank you very much. The next question is, Trehalose is available as an OTC supplement like TUDCA. Although the quality of such products is often uncertain, is this option available for extension of regimen E?
Yeah, that's a really good question. So trehalose is it's a disaccharide, so it's a sugar molecule, and you're right, it's available as an oral over the counter, but we all have this enzyme in our stomachs that digests it, so you can't you can take it, but you will never get enough into the blood and then into the brain. So it really has to be given IV. So in the regimen E, it's a weekly IV infusion. So unfortunately, the oral won't work. I think because the first act for ALS expanded access program was for Selos, the trehalose drug. We're hoping that the people who are not eligible for trials could do that, as well as the people from the OLE could go into that. And we have requested to have more support from the NIH to grow that even more. We expect to have the results of regimen E sometime, I think, late fall. We're trying to work even faster to get it early fall. I think if it's positive, then the company would probably be able to, reopen the open label and grow that as well.
Thank you. We have about 5 questions left. So our next question is, what metrics are being used to assess whether F and G reach the primary endpoint of each study?
Yes, so the primary endpoint is actually the same for F and G, in the sense that we're measuring the progression of the disease by measuring, we've actually a very innovative statistical model measuring the ALSFRS-R and how it changes over 24 weeks. That's really the primary metrics. And then we have a number of other supportive endpoints or outcomes. And the reason for that is that we need to characterize, we always want to characterize the full effect of the drug in people living with ALS. So we're measuring, for example, muscle strength, respiratory function. We're measuring patient-reported outcomes, such as the Rhodes questionnaire, which is a questionnaire reported by people living with ALS, or other patient-reported outcomes, for example, quality of life and other metrics like that. Obviously, as discussed earlier, we have a big interest in biomarkers. And so while they're not the primary endpoint, we're trying to learn as much as possible, so that hopefully in the future, there will be at the level where neurofilament is today. And so right now they are more in a supportive role, so to speak, but really they could become more prominent as we learn more about them.
All right, perfect. Next question is, how do you identify subsets of ALS patients with different causes and forms that will result in different reactions to medications?
Yeah, that's an excellent question. So we do it. We look for a couple different ways. One is we do ask people if they can provide blood for DNA, and we do whole genome sequencing. So look if there's any kind of genetic background that might define a different subset. We also look at some of these digital and fluid biomarkers, and then we look at some clinical characteristics. But I think we need, in general, more tools to be able to do that. I know that's a lot of what everything ALS is working on, because we don't yet have anywhere near what like oncology has to be able to subset people on different biology and different response to drugs. But we need to get there.
Thank you. Our next question is, how often are you collecting NFL samples, and when will you have enough samples to pre-publish some results?
Yeah, so we definitely have a lot of samples already, and we are definitely really working hard on the analysis and hopefully publications later this year. We're working on them right now. So we collect neurofilament at 4 time points during the placebo-controlled trial, and then 2 additional time points during the active treatment extension. So baseline week 8, 16, 24, and then a week 8 and 16 again of the active treatment extension.
Thank you. Our next question is, what percentage of those patients that receive a placebo intervention showed therapeutic improvement or mitigation of progression?
That's a good question. I don't want to misspeak, but I don't think that we saw in the A through D anyone in the placebo group get better. We certainly have some people who with slow, very slow. But I'd say this all with about 96% confidence. I think we have to go back and look at that. So good question.
Thank you. And our last question is not so much a question, but we did see that a lot of members in the chat have said that in their cities, such as San Antonio or Mayo Rochester or in Connecticut, they had some difficulty either connecting with the site that has the Healey trial, or that it's not active yet, but it says it is on the website. So do you have any information on where they can reach out and get more information so that they can be a part of the Healey trial?
Absolutely. First of all, thank you for your interest. So for regimen F, we have 50 of our 70 sites activated, and we're getting very close on the other 20. I'd say of those 20, probably 10 are very close in the next month. Catherine Small and Allison Boulat would be the go-to people. They're our navigators for the trial, and they'll know, for example, if they'll know if San Antonio's maybe 2 or 3 weeks away, but or longer, and then they can actually help contact another site in Texas or close by to see if they could see you sooner. And they do that for many people. That would probably be the best set. The question is why are some sites activated now and some are take longer? And this is a lot of internal, because some sites have had a lot of turnover of their staff, so they might not be able to get activated. But our goal is that all sites are activated within the first 2 months. I think we got to 50 of them in the first 2 or 3 months, and we have some stragglers, but we're working on it.
All right. Thank you so much. That was a lovely Q&A session. We really appreciate your time and all of your hard work and answers. Thank you so much. I'll pass it back to Indu and McFinn. We do have a poll today, just a small feedback poll we're going to run to see how everyone loves the session and if there's anything that you guys recommend, but we can run that maybe during McFinn's time.
Thank you for having us.
No problem. Thank you for being here.
Well, thank you, folks. How do you like that? Right to your living room. You didn't even have to go traveling. All you had to do was open your heart, your minds, and your Zoom program, and we are a family again. Every 7 days, each of us have an opportunity to share what we've discovered over those past 7 days, because in one day, you can come up with a trick or tip that's going to make somebody else's life really easier. So we're all important. Each one of us is a detective. And all I want to do is say to our 2 presenters this evening, your time is precious and we are precious. So we match. So thank you so much. It's really an honor to feel like we've attracted your attention. And everybody here, I'm sure, is extremely thankful that we can be part of your team and that you're part of our team. So folks, it's a win-win situation. Welcome to today. And today is one of the most important days of your life. And so please, before you go to sleep tonight, look in that mirror and say, I love you. Let's beat this. It's really an attitude adjustment that will keep us moving on. So once again, thank you so much to our two presenters and to everyone here. I mean, each one of you are a presenter. Believe it. Each one of you has a little something that somebody else just might need. And so each one of us are precious. Let's remember, there's always two of us, that one that has ALS and that one of us that is whole and complete, sitting inside, hoping for the day when we hear ALS is over. So with that, Indu, is there anything you'd like to say before we go into our open forum?
Well, thank you very much, Merritt and Sabrina, for joining us this evening and sharing. And I know how hard you guys work. And, this is, like we say, it's an army here. And we're also thankful that we have, experts like you in our army. So thank you very much for everything you do and the passion that you bring.
Thank you.
Thank you.
Bye-bye.
Thank you.
Bye-bye.
Bye-bye.
And we're speaking for everybody here on the show tonight. So everybody, you are so important that we have some of the most important people in the United States here visiting with us tonight, bringing everything that they've collected over so many years. And that's the real gift, is that you two are dedicated to this. And I hope you go to sleep at night and feel that all of these people out here are cheering you on.
It really does feel like a close community. Even though we might not have met each other in person, we are all there together. The same person, same purpose and same passion.
Yes.
Thank you.
Thank you so much.
Bye.
All right, everybody.
Thank
Thank you so much.